Welcome to this lecture series on eukaryotic gene expression, basics and benefits. This is the 27th lecture in this series. In the last few lectures, we have been discussing the role of gene expression during embryonic development. We have been using Drosophila as a model system. I have tried to explain how maternal genes as well as zygotic genes and their differential expression ultimately is responsible for the development of the single cell zygote into a multicellular organism, how spatial and temporal expression of genes play a very important role in the regulation of gene expression at different stages during development. And we discussed things like maternal effect genes, then segmentation genes, then we talked about homeotic genes, and how all the expression of these various genes during various stages of development ultimately results in the development of an adult organism. The point I made in the last 3 lectures in trying to understand the role of gene expression during development is the DNA sequence as well as the spatial and temporal expression of the various genes plays a very important role during embryonic development. But I do not want to give an impression that it is only the DNA sequence and it is only the transcription of this sequence information from the gene into messenger RNA and ultimately translation to protein that alone is responsible for the development of an adult organism from a fertilized egg. What I would like to tell you today is that in addition to this genetic basis, in addition to the DNA sequence information, epigenetics also plays a very important role in the regulation of embryonic development. So, let us try to understand what is this epigenetic regulation of gene expression during embryonic development and how important it is for the proper development of the embryo. Now, development of a multicellular organism is not only determined by the DNA sequence, but also epigenetically through DNA methylation and post-translation modifications of histones. Now, in some of the early classes when we discussed about the role of chromatin in the regulation of gene expression, I had extensively told you how the histone post-translation modifications as well as DNA methylation play a very important role in the regulation of gene expression, and in fact based on this we had discussed about what is called as a histone code and epigenetic code and so on and so forth. So, what we will discuss today is that how is this modification of histones as well as DNA methylation play a very important role in the regulation of embryonic development as well. Epigenetic events influence the expression of several genes during embryonic development. If we take an example, the embryonic stem cells, that is, these are the cells at the stage of blastula which are totipotent and which have the capability to develop into a full-fledged organism. The embryonic stem cells, if you take the embryonic stem cells, active gene expression markers are usually found in pluripotent genes, whereas repressive markers are found in the lineage specific genes, clearly indicating that the epigenetic markers are pretty different in different cells of the embryo. Thus, different cell types and their fates can be defined by their epigenetic and gene expression profiles. Now, as I just mentioned, we had extensive discussions about the various post-translation modifications histones undergo and how these post-translation modifications play a very important role in the regulation of gene expression. For example, we had discussed how acetylation of histones by a group of enzymes called histone acetyl transferases can result in the activation of gene expression or is a kind of a fingerprint for actively transcribed genes, whereas when histones are deacetylated it usually signals repression of gene expression. In addition to this acetylation and deacetylation, histones also undergo several modifications like phosphorylation, methylation, ubiquitination and so on and so forth. And based on all these studies, people have actually identified what are called the signature motifs. That is, today I can say if in the promoter region of a gene, if I find a histone who H4 whose lysine residue 6 is acetylated, then I can say this is likely to be actively transcribed. So, this is what is called as a histone code hypothesis. So, based on studying a number of genes and the kind of histone modification that are associated with the expression status of these genes, people have now developed what is called as a histone code hypothesis. Some of the examples I have shown here. For example, if you have acetylation of histone H3 or H4, usually it signifies that these genes are likely to be transcribed very actively. Similarly, deacetylation of histones usually signals inactive genes, and in the nucleus if you look at the heterochromatin region which basically contains inactive genes, usually histones remain deacetylated in the heterochromatin regions of the nucleus. Similarly, methylation of the lysine 4 residue of H3 usually associated with actively transcribed genes, whereas methylation of the lysine 9 residue of the histone H3 usually signifies that this is involved in gene silencing, and genes in this region are not likely to be expressed very actively. So, this based on these kinds of studies we have discussed what is called as histone code hypothesis, and after studying the histone code hypothesis we had also discussed, in addition to histone modifications, DNA modification, especially the methylation of cytosine, also plays a very important role in the regulation of gene expression. We had discussed how 5-methylcytosine, because of its importance in regulation of gene expression, has now come to be known as the 5th base in addition to adenine, thymine, guanine and cytosine. And together the histone code and the DNA methylation patterns constitute what is called as a epigenetic code. So, what we have learned so far in our earlier lectures is that even without altering the DNA sequence information, you can alter gene expression by either bringing about post translational modification of histones or by altering the methylation pattern of the DNA. This is what is called as the epigenetic regulation of gene expression. Now, let us try to understand how does this DNA methylation or demethylation of cytosine residues of DNA regulate gene expression during development. Now, let us try to understand what kind of methylation changes takes place during development both in the maternal as well as the paternal genome. Now, let us try to start our discussion just before fertilization. What is the status of this methylation status as well as the genome status in the sperm as well as the oocyte. Now, prior to fertilization, mammalian gametes are at different stages of cell cycle and their genomes are organized very differently. If you look at the egg, for example, the egg is meiotically arrested at metaphase 2, resulting in a diploid genome that is packaged with histones. Okay. So, the egg does not undergo complete meiosis, the meiosis is arrested at the metaphase 2 stage. The mature sperm on the other hand completes the meiosis, but their haploid genomes are packaged with protamines instead of histones. So, during spermiogenesis you can clearly see most of the histones are replaced by another distinctly set of DNA binding proteins called as the protamines. So, histones no longer are present in the sperms. When a sperm penetrates the zona pellucida to fertilize the egg, that is during time of fertilization, both the gametes undergo very rapid changes. So, you had the oocyte in which the meiosis was not completed, it was at in metaphase stage. You had a sperm in which the DNA is highly condensed, but most of the histones are replaced by what are called as protamines, and also they have what are called as some testis specific histone variants replace the normal somatic histones in the present in the other somatic cells. But the moment the sperm penetrates the egg, rapid changes are take place not only in the sperm nucleus but also in the oocyte nucleus. What happens to the oocyte? The egg completes its second meiosis, resulting in the extrusion of one copy of the genome as polar body. The sperm reorganizes its genomic DNA by replacing protamines with histones. So, you can see during spermiogenesis the histones are replaced by protamines, and this tightly condensed highly condensed chromatin on the sperm DNA. Now, after fertilization the protamines are again replaced by histones in the fertilized egg in the case of the sperm. Now, around 4 to 8 hours after fertilization, especially after the protamine histone exchange, that is when the protamines in the sperm nucleus are replaced by the histones, the sperm derived paternal pronucleus undergoes what is called as a genome wide DNA demethylation, and this is completed before the first cell division. So, the methylated cytosines in the sperm DNA are removed very actively, and the sperm DNA undergoes very extensive demethylation within about 4 to 5 hours after fertilization. So, first thing that happens after the entry of the sperm into the egg nucleus into the egg is the protamines are replaced by histones, and then the sperm DNA undergoes extensive demethylation. The paternal DNA, this demethylated DNA remains demethylated even after multiple rounds of cell division, after the fertilized egg divides several times. So, specific cytoplasmic factors which are present in the fertilized egg are responsible for selective demethylation of the paternal DNA. Remember, you have the oocyte DNA and you have the sperm DNA. Only the sperm DNA undergoes selectively demethylation within hours after fertilization. Thus, although both maternal and paternal DNA are exposed to the same cytoplasmic factors, only the paternal but not the maternal DNA is demethylated. So, you can see already the maternal nucleus and the paternal nucleus are already driven. The DNA inherited from the father, the DNA inherited from the mother, they are already very different. One of them undergoes extensive demethylation, other does not. One of them, the protamines are replaced by histones, but other remains the same. So, the paternal maternal nuclei are not the same. So, this may be due to a mechanism that protects the maternal genome from this way of demethylation, or due to a putative DNA demethylase that specifically recruited to the paternal genome. So, what is the mechanism? You have two nuclei, you have DNA in both of them, but only one of them the DNA is extensively demethylated, but other is not. So, there is some mechanism by which there are certain factors present in the egg cytoplasm, they recognize only the paternal DNA and demethylate only the paternal DNA and not the maternal DNA. So, in addition to the replacement of protamines with histones in the case of the sperm DNA, specific histone variants are also deposited in the paternal pronucleus. So, not only protamines are replaced by histones immediately after fertilization, certain specific variants of histones are also deposited in the sperm DNA. One of the examples is histone H3.3. This particular variant of the histone is preferentially deposited in the paternal pronucleus, and it is suggested it is differences such as these, that is replacement of protamines by histones and selective addition of only the histone variants like H3.3 only to the paternal DNA but not in the maternal. These may serve as actually signals for these demethylases present in the oocyte cytoplasm to selectively demethylate only the paternal genome but not the maternal genome. In addition to certain histone modifications, paternal or maternal specific, in addition certain histone DNA modifications are also paternal or maternal specific. For example, methylation, dimethylation and trimethylation of the histone H3 at lysine 27 as well as at lysine 9 are seen only in the maternal pronucleus of the zygotes but not in the paternal nucleus. The point I am trying to make is that although you have the DNA inherited from the sperm and the DNA of the oocyte, although the genetic component in terms of DNA sequence is the same, more or less the same in both, but you can see some of these epigenetic changes are quite different. In the case of the sperm DNA, the DNA undergoes extensive demethylation, whereas the oocyte nucleus does not undergo that. The same way, certain specific variants of histones are added only to the paternal nucleus but not to the maternal genome. In addition, specific histone modifications take place only in one of them but not the other. So, already you can see the epigenetic signatures of the maternal paternal genomes are pretty altered very early during embryonic development, in fact immediately after the fertilization. So, the maternal genome may use a very protective mechanism against demethylation by carrying certain specific histone variants or histone modifications. So, because of these differences in the epigenetic nature of these two genomes, one of them undergoes extensive demethylation, another do not. So, people started looking at and see what kind of factors actually regulate this differential demethylation of these two genomes, and extensive research is going on. And one of the interesting component that has come out recently is that it has been reported that certain non-histone factors present in the oocyte might protect the maternal genome from demethylation. For example, zygotes lacking a maternal effect gene known as STELA or DPPA3 or PGC7 exhibited demethylation of both pronuclei, although how STELA protects the maternal genome from demethylation remains to be determined. So, some of the studies, in fact this is a very nice paper which was published in Nature Cell Biology in 2006 entitled PGC7 or STELA protects against DNA demethylation in early embryogenesis. That means this is a maternal effect gene, that means this gene is already transcribed and the RNA is already stored in the oocyte before to fertilization. But the product, the protein arising out of this maternal RNA is known as the STELA, and if you have this STELA, the maternal genome does not undergo methylation. But if we now delete, or if we make a mutation, or if you have a mouse in which this particular gene is knocked out, then both maternal as well as paternal genomes undergo demethylation, indicating that one of the cytoplasmic factors that may protect the maternal genome from demethylation is this protein called as STELA. But the exact mechanism of which this protein is acting is still not very well understood. So, Let us now try to understand what is exactly the significance of this DNA demethylation and DNA methylation during embryonic development. DNA demethylation involves removal of the methyl group from the 5-methyl cytosine. As I said, immediately after fertilization the paternal genome undergoes extensive demethylation. This demethylation can take place by two mechanisms: active demethylation or passive demethylation. In the case of active demethylation, it actually involves removal of the methyl group from the 5-methyl cytosine. That means a demethylase has to be expressed in the oocyte cytoplasm after fertilization, and somehow this demethylase will remove the methyl group from the cytosines of only the paternal genome but not the maternal genome. The other alternative is that there can be actually inhibition of the DNA methyl transferase 1, therefore this methylation cannot take place. So this is called as a passive demethylation. So when a DNA methyl transferase is not expressed, so methylation is not possible, and therefore there is no methyl cytosine residues present. Now even though DNA methylation contributes to stable, long term and heritable gene silencing, we had already discussed in the previous classes how once the DNA is methylated, the methyl cytosine attract proteins like the MECP2, and these MECP2 in turn recognize specific histone deacylases, and then histone deacylases come and deacylate histones, and this is how DNA methylation leading to histone deacylation ultimately results in the repression of transcription, or results in inactivation of the gene expression. So the DNA methylation actually can lead to gene silencing, usually involving histone deacylation. So the DNA demethylation can occur at two different levels. One is genome-wide demethylation, which occurs at specific times during early embryonic development. As I had just now mentioned, the entire paternal genome, most of the cytosines get demethylated. On the other hand, it can also be gene-specific demethylation, which usually occurs in somatic cells responding to specific signals. So many of the epigenetic changes of regulation of gene expression we studied in our earlier classes belong to the second category, where we have discussed how specific transcription factors, when they go and bind to promoter regions by either recruiting DNA methyl transferases, can methylate DNA, and this in turn can attract histone deacylases, and therefore those genes can get inactivated. This is called as gene specific methylation or demethylation. But what we are discussing during the early embryonic development is that the entire genome, most of the cytosines getting demethylated. So during embryonic development, some genomic regions are resistant to demethylation. Now when I said the entire paternal genome undergoes demethylation, it is not that every methyl cytosine is, the methyl group is removed. There are certain regions in these genomes, either paternal or maternal, they are protected from undergoing this kind of a demethylation. So there is a global wave of demethylation going on. You can see how interesting the whole scenario is. Immediately after fertilization, selectively the paternal genome gets demethylated, but now I am telling not all regions of the paternal genome get demethylated. There are certain regions within the paternal genome which are protected from undergoing this demethylation. These regions include what is called as imprinting control regions, intracisternal A particles, these are nothing but certain transposons, and centric and pericentric heterochromatin, that is the chromatin or the DNA in and around the centromeric region are also protected from undergoing this kind of a demethylation. So there are certain regions which are protected from this global wave of demethylation that happens immediately after fertilization. Now now the paternal genome got demethylated. Now how does it get undergo methylation now? So the egg now starts dividing, and as the egg did, what kind of enzymes are involved in the methylation of this demethylated DNA? Specific patterns of DNA methylation are re-established during early embryonic development by the action of DNA methyl transferase and demethylase enzymes. This is a very important aspect. I would like to spend some more time to emphasize this point because it has a lot of implications. What I am trying to tell you now is that as soon as the fertilization is over, the genomes of paternal maternal undergo extensive epigenetic changes. It could be histone modification, it could be replacement of protamines with histones, or it could be addition of histone variants. In addition, selective demethylation of the paternal genomes, all these things take place. Now once the demethylation has taken place, a new methylation pattern is established during the embryonic development. This has a lot of implications. As we go along the lecture, it will become very clear. Today people are trying to do what is called as a nuclear cloning, okay, wherein you take the nucleus from a skin cell, that is the adult diploid cell, and put this nucleus inside a oocyte which has been enucleated, and see whether this adult nucleus can now trigger embryonic development, and can you produce an embryo, and can an adult develop from such kind of a the thing. This is what is called as a cloning, okay, like the Dolly sheep was produced by these kinds of methods. But what I am trying to tell you is that the epigenetic program of an adult differentiated nucleus is not exactly the same as that of either the sperm nucleus or the oocyte nuclei. So, this is the reason, although the DNA sequence information may be same, because of this difference in the epigenetic signatures of these, that is why the failure rate of producing these kind of offspring from these kinds of somatic nuclear transplantations are very low, because the epigenetic reprogramming that takes place in early embryonic development may not be the same when you take an adult differentiated nucleus and put inside a oocyte. That is very different from that of a sperm versus egg fusing together and forming a diploid nucleus. Now, the enzymes that do this DNA methylation is called DNA methyltransferases, and in mammals the DNA methylation occurs predominantly the CpG residues. We have discussed these things quite a bit in our earlier classes, but interestingly in the case of plants, DNA methylation can occur at cytosine bases in many other sequence contexts, whereas in animals predominantly it is the dinucleotide CpG which is the substrate for DNA methylation. The cytosine in the CpG repeats get methylated, and the enzymes responsible for this DNA methylation are known as DNMTs or DNA methyltransferases. There are two types of DNA methyltransferases: de novo DNA methyltransferases and maintenance DNA methyltransferases. Now, the DNA methylation patterns are initially established by de novo DNA methyltransferases. There are two such enzymes called DNMT3A and DNMT3B during the blastocyst stage of the embryonic development. As I said, the genome undergoes extensive demethylation immediately after fertilization, and it is this DNMT3A and DNM3 which then again re-establish the methylation pattern of the zygotic nucleus. These methyl markers are then faithfully maintained during subsequent cell divisions by the action of maintenance methyltransferases called DNMT1, which has a preference for hemimethylated DNA. Let me try to explain these two aspects using say specific cartoons. What exactly you mean by de novo DNA methyltransferases and maintenance methyltransferases? So, there are three DNA methyltransferases. One is called DNMT1, which is a maintenance methyltransferase, and DNMT3A and DNM3B, which are called as de novo methyltransferases, and 3A and 3B are highly expressed in the embryo implantation when waves of de novo methylation occur in the genome. Let us try to understand what exactly happens using a cartoon now. This is what happens. As I said, let us assume that this is a DNA from a sperm, and you can see the DNA has now been completely demethylated by the action of the demethylases immediately after fertilization, and this demethylated DNA now acts as substrate for two enzymes DNMT3A and DNMT3B. These are called as de novo demethylases, and they now add methyl groups to cytosine residues of this, let us say for example a sperm DNA. So, during early development, methylation patterns are initially re-established by the de novo DNA methyltransferases DNMT3A and DNMT3B. Now, then as the zygotic nucleus divides, the DNA replicates and DNA also divides. So, the daughter strands are formed, and therefore what you get is a hemimethylated DNA. So, the methylated DNA, the two strands which are then go, now one strand goes to each one of the daughter strands, parental strands, and therefore the newly formed daughter strands remain unmethylated. So, what you get is a hemimethylated DNA after first round of replication. This hemimethylated DNA now becomes a substrate for the next enzyme DNMT1. Okay. So, the methylation is first re-established by the de novo methyltransferases 3A and 3B. Once the specific methylation pattern is established by them, this pattern is maintained during successive cell divisions by this maintenance methyltransferase. For example, in a promoter region in a gene, for example, let us there are 10 cytosine residues, but only 5 are methylated by the DNMT3 and DNMT3B. The same 5 residues will continue to be methylated by the DNMT1 through the subsequent cell division. So, the methylation signature is maintained by the maintenance DNA methyltransferase. So, the hemimethylated DNA is a better substrate for this enzyme than the completely methylated DNA, and therefore the other daughter strands get methylated, and therefore the hemimethylated DNA now becomes fully methylated. So, DNMT1 acts on the hemimethylated DNA and methylates the newly synthesized DNA, and thus maintains the methylation signature of the DNA during embryonic development. Now, DNA methylation. So, what I told you so far is that first there is a demethylation, and then there is a remethylation by both de novo methyltransferases as well as maintenance methyltransferase. So, once this methylation signature is established after fertilization in the zygotic nucleus, let us now try to understand what is the significance of this DNA methylation, what kind of functions that it regulates. DNA methylation plays a very important role in differential gene expression during embryonic development. I am going to discuss today only two examples. One is called genome imprinting, another is called X chromosome inactivation. Let us take these two processes and see how DNA methylation plays a very important role in these two processes. Now, what is imprinting? Imprinting is nothing but selective inactivation of maternal or paternal alleles such that only the paternal or maternal allele is expressed. Let me repeat. Imprinting is nothing but selective inactivation of the maternal or paternal allele such that only one of these alleles is expressed during the development. Let us elaborate a little bit what exactly this is. Now, in a diploid organisms, the somatic cells possess two copies of the genome, because one comes from the father, one comes from the mother. Now, each autosomal gene therefore is represented by two copies or alleles with one copy inherited from each parent at the time of fertilization. So, every gene has two alleles, one comes from the father and one comes from the mother. Now, in majority of the genes, let us say for example, the humans we have about anywhere between 30 to 50,000 genes, 99% of them or even more than that, both the alleles are expressed. That is, the allele inherited from the sperm as well as allele inherited from the oocyte, both are expressed. But in some cases, a very small proportion, about 1% of the total genes, only one of the alleles, either paternal or maternal, is expressed such that gene expression occurs from only one allele. So, you can see, although we have two alleles for every gene, one coming from the mother and one coming from the father, although 99% of these genes, both the chromosomes, so both the alleles are expressed, about 1% of the genes, either a paternal allele is expressed or maternal allele is expressed, and this is what is known as imprinting. So, selective inactivation of one of the alleles is known as imprinting. The term imprinting was first used to describe events in the insect Pseudococcus snipae, what popularly known as mealybugs. Now, in these insects, both male and female develop from a fertilized egg. Okay? Once fertilization is there, both male and female develop from a fertilized egg. However, in females, all chromosomes remain euchromatic and are functional. There is no inactivation. Whereas, in the embryos which are destined to become males, one haploid set of chromosomes become heterochromatinized after the 6th cleavage division and remain so in most of the tissues. So, the males, one set of chromosomes get heterochromatinized or get inactivated. So, there will be no expression of genes from this particular set of chromosomes, although therefore, although they possess two sets of chromosomes, one from the paternal and one from the maternal side, functionally they are haploid, because only one set of genes are getting, one set of alleles are going to get expressed. So, in insects, this kind of a imprinting leading to expression of only one set of alleles describes the silencing of paternal genome in males and thus therefore is involved in sex determination. So, if that fertilized egg has to develop into males, the one set of alleles get inactivated, therefore functionally becomes a haploid and such eggs develop into males, whereas in those eggs in which both alleles are active, they develop into females. So, this kind of an imprinting that takes place in the insects actually determines the sex of the fertilized egg, whether it should develop into a male or it develop into a female. The fact that both maternal and paternal genomes are not exactly same became evident from nuclear transplantation experiments in mouse zygotes in the early 1980s. So, all that I told you so far clearly indicates that although the number of chromosomes and the gene sequence, everything is more or less same in the sperm as well as the oocyte, all the information I told you very clearly tell that as far as the epigenetic signatures are concerned, it is not the same, they are different. How did they prove that? In vast majority of the mouse parthenogenomes or gynogenomes, that is, these are the ones of eggs which contain the two maternal or the egg genomes, no sperm genome, or those which contain two sperm genomes but no oocyte genome, they all die before the blastocyst or implantation stage. So, if the genome from the oocyte and the genome from the sperm are exactly the same, if you replace the sperm nucleus and instead put one more oocyte nucleus, it should still develop the same. So, these are called as the gynogenomes, that is, we have genome from two eggs, or you take out the oocyte nucleus and introduce two sperm genomes. So, these are called as the androgenomes. But when you do these things, such kind of fertilized eggs, for such kind of eggs, this is called as parthenogenesis, they do not develop at all. So, you need to have a maternally inherited genome and a paternally inherited genome for proper development, clearly indicating that they are not exactly the same. These studies indicated that normal development requires the contribution of both maternal and paternal genomes. If you have either of them, development is abnormal. So, parthenogenetic or gynogenetic embryos have twice the normal expression of level of maternally derived genes and lack the expression of paternally expressed genes, while the reverse is true for the androgenetic embryos. So, if you take some of these embryos, which although they die at an early stage, but at those early stages, if you look for the expression of genes, there are a lot of abnormalities in the gene expression. Okay, some of the genes are expressed twice, and some of the genes which are normally expressed from the parental allele, they are not expressed at all. So, in mammals, genomic this is all in the insects. Okay, in the insects there is complete activation of one set of alleles, and then early studies have clearly said that if you now put either 2 oocyte genomes or 2 sperm genomes, it results in abnormal development and abnormal gene expression. So, in mammals, genomic imprinting describes the processes involved in introducing functional inequality between 2 parental alleles of genes. Okay, there are some genes which are not expressed in double dose, they are expressed only in single dose. We will understand better if you now give us 1 or 2 examples of this imprinting and then tell you how exactly this imprinting takes place. 2 of the very well studied imprinted genomes or imprinted genes in humans are, one is called as the H19, another is called as insulin-like growth factor 2 or IGF2. Now, interestingly, although the H19 gene is present in both paternal chromosomes as well as maternal chromosomes, the H19 is expressed only from the maternal chromosome. That is, only the maternal allele of the H19 is expressed during embryonic development in the adult. Similarly, the IGF2, only the paternal allele is expressed, but not the maternal allele. So, you can see these are reciprocal. The H19, always the maternal allele is expressed, the paternal allele is not expressed, whereas in the case of IGF2, it is the paternal allele which get expressed, but not the maternal allele. Like this, in the mammalian gene, at least 100 such imprinted genes have been identified so far. In these cases, although both the alleles are present, only one of the alleles will be expressed at any given time. So, it turns out these genes are imprinted by selective methylation of one of the alleles. So, this kind of imprinting, the basic mechanism that is responsible for the selective inactivation of one of these alleles is nothing but DNA methylation. Let us now try to understand how exactly this takes place. Now, H19 codes for a long non-coding RNA that is transcribed only from the maternally inherited allele, and the paternal H19 allele is not expressed. Now, I will not complicate you at this time what is called a non-coding RNA. We will discuss this in a later lectures. A non-coding RNA nothing but it is an RNA which is not translated into a protein. That is, the functional molecule is an RNA and not the protein. In fact, there are now several non-coding RNAs in addition to ribosomal RNA and transfer RNA which are also non-coding RNAs. These are well-known non-coding RNAs. Studies in the last 2 decades have led to the identification of several such non-coding RNAs. We will discuss some of these non-coding RNAs at later stages of our lecture series. Now, a major function of this H19 is to regulate the expression of another gene known as IGF2. And let us see how exactly this takes place. Now, the regulatory elements which control imprinting, that is, which are responsible for selective expression of either the maternal or paternal allele, are known as imprinting control regions and differentially methylated regions. The cis-acting regulatory elements which are responsible for this imprinting or expression of only one allele or the other are known as imprinting control regions or ICRs or DMRs or differentially methylated regions. Now, let us take H19 for example. The H19 contains a differentially methylated region that also acts as a imprinting control region or ICR. The ICR is differentially methylated at the CPG residues according to the parental inheritance. So, there are CPG sequences within this internal control region or the imprinting control regions of the H19 gene, and this region or the ICR, the cytosine regions undergo differential methylation depending upon whether it is the paternal allele or the maternal allele. Usually, the paternal copy of H19 is methylated and silent, whereas the maternal copy is hypomethylated or unmethylated and therefore expressed in the offspring. So, you can see you have 2 alleles, one coming from the mother and one coming from the father. In the case of H19, the maternal allele is hypomethylated and therefore it is expressed, whereas the paternal allele is methylated and therefore it cannot get expressed. So, when H19 promoter DNA is methylated, its expression is shut off. As we have studied earlier, DNA methylation results in the attraction of the histone deacylases, which has to deacylate histones, and therefore it can result in condensation of chromatin and therefore inactivation of gene expression. So, when H19 promoter DNA is methylated, its expression is shut off. But very interestingly, when H19 promoter expression is shut off, the same time the expression of IGF2, which is a neighboring gene, H19 and IGF2 are present side by side in the same chromosome. When H19 expression is shut off, the expression of IGF2 is stimulated or is turned on. Now, let us try to understand how repressing the transcription of one gene results in the activation of expression of another gene. How does it take place? Very simple. There are specific cis-acting elements called as insulators, which play a very important role in this differential expression of gene expression. That means, when the expression of H19 is turned off, the expression of the neighboring gene IGF2 is turned on, and certain specific cis-acting elements or promoter elements called insulators play a very important role in this differential expression of these two neighboring genes. I have now put a cartoon here to explain, although it is a very complicated phenomenon. I try to explain it in a very simple manner. Let us now understand a mechanism by which this actually takes place. Now, this is the human chromosome 11. In the human chromosome 11, both the H19 gene and IGF2 gene are present side by side. They are neighboring genes. Now, the H19 has an enhancer sequence at the 3' end of H19 gene. Usually enhancers are present at the 5' end, but in this case the enhancer is present at the 3' end, and enhancers can act in a orientation independent manner. Now, in the maternal chromosome, where the H19 gene is normally expressed, transcription factors recognizes this enhancer sequence and they bind to it, and therefore this transcription factor now activates transcription from the transcription start site, and therefore H19 RNA is made. Okay. But this transcription factor cannot activate the expression of IGF2 because there is a sequence called insulator sequence, and to this insulator sequence a protein called CTCF binds. So, when CTCF binds to this insulator motif, this transcription factor can only activate the expression of H19 and it cannot activate the expression of the IGF2 promoter. Therefore, in the maternal chromosome only the H19 gene is expressed, but the IGF2 gene is not expressed. Okay. Now, let us see what happens to the paternal chromosome. In the paternal chromosome, the insulator region in the H19 region, the insulator sequence near the H19 region is methylated, and when this gets methylated, now this CTCF protein can no longer bind to the insulator sequence, and therefore this insulator can no longer prevent the activation by this transcription factor of the IGF2 promoter. Now, since this H19 promoter is methylated, H19 gene cannot be transcribed because now the histones are deacylated here and therefore the chromatin is condensed. But however, because the insulator sequence is not bound by CTCF, now the transcription factor which is binding to the 3' region of H19 enhancer, the activity it can now, there is no barrier here, this insulator barrier is now removed, therefore this now goes and activates the expression of IGF2, and that is how IGF2 now gets expressed. So, you can see methylation of this insulator sequence removes the barrier, and therefore a transcription factor binding to the H19 enhancer can now activate the transcription of the IGF2 gene. Whereas, when the H19 promoter and insulator sequence is not methylated, a protein called CTCF binds to the insulator, and therefore it acts as a barrier and prevents the transcription factor from activating the transcription of the IGF2 sequence. And you can see how nicely these two now reciprocally regulate the expression of each other. So, that is why whenever H19 is expressed, IGF2 is not expressed, and whenever IGF2 is expressed, H19 is not expressed. So, it is a wonderful example how epigenetic regulation takes regulate the expression of two different genes. What is the application of this? As I said, this course, whenever possible, I always try to give applications to see how understanding some of these basic mechanisms have led to the benefit of mankind. Now, one of the major problems in the areas like gene therapy, which again we are going to discuss a few classes later, is that when I want to treat a genetic disorder, when I want to express my gene of interest, when I interest the gene along with the promoter into the cells, the DNA goes inside and integrates into the genome, but expresses only for a short time and then it gets inactivated. It could be either because if the DNA goes into heterochromatin regions, or it could be undergo histone deacylation may take place in promoter region. Whatever the reason, the foreign DNA does not get expressed very highly. This is one of the major problems in a number of applications like gene therapy and so on and so forth. But now what people have now found out is that if you now take this gene and if you now construct the vector in such a way that you now put insulators both upstream and downstream of this gene sequence. If you now put that, then the chances of this kind of a silencing effect acting on this your gene of interest is very, very low. So a gene inserted at random into mammalian genome is often silenced. This is one of the major problems of gene therapy. We will discuss this again when we come to gene therapy. But if you now place this kind of insulator sequence on either side of the gene and then introduce this gene into the nucleus, these insulator sequence will prevent this silencing effect onto the transgene, and therefore your transgene can be protected from gene silencing, and therefore your gene can be expressed for a very long time. This has tremendous implication in the area of gene therapy. We will discuss this in detail when we talk about gene therapy in the later classes. In addition to this mechanism, what I told you in the case of H19 and IGF2, there are a number of mechanisms that are involved in imprinting. For example, expression of certain non-coding RNAs such as AIR on mouse chromosome 17, KCNQ1 OT1 on human chromosome 11p, they all play a very important role in genome imprinting. What I am trying to say is that recent studies indicate that many non-coding RNAs, that is, these are RNAs which look like messenger RNAs, but they do not code for any proteins, but they seem to have very important regulatory functions, including imprinting. And such RNAs seem to play a very, very important role in the chromosome genome imprinting. I will not go into the details. If you are interested, you can go through this article, the AIR non-coding RNA and imprinted cis-acting silencing transcript, and then study how exactly these non-coding RNAs regulate gene expression. Now, this imprinting is very, very important, and as you can see here, abnormal imprinting during development results in a number of genetic disorders like Beckwith-Wiedemann syndrome, Silver-Russell syndrome, Angelman syndrome, Prader-Willi syndrome. So if this imprinting does not take place properly and this maternal versus paternal gene activation is not done properly, it can result in a number of genetic disorders. There are many such interesting examples of differential gene regulation involving imprinting and so on and so forth during embryonic development. Another interesting example is what are called as there are certain cis-acting elements known as locus control regions. So regulation of certain genes during development requires what are called as the locus control regions or LCR. Now, the best example to understand this locus control region is the expression of the globin genes during development. Human and mouse globin genes are clustered in the genome and are differentially expressed at different stages of development. Now, this is the globin gene cluster. What I have shown as a clear, there is what is called the epsilon gene, gamma, delta, and beta. The beta is finally expressed in the adult, whereas during embryonic development, these other variants of this globin gene is expressed. Only in the adult, the beta globin is expressed, but they all come from the same locus. So there is a common cis-acting regulatory element called as local control region, which differentially activates these genes depending upon the various stages of development. So a group of regulatory elements collectively known as locus control region present 30 to 50 kb upstream of the cluster of genes plays a very, very important role in this differential activation of these various genes in the beta globin locus. This locus control region binds to regulatory proteins and causes chromatin structure to open up, allowing access to an array of regulators that control the expression of individual genes in defined order. I will not again go into the details, it is a very complicated subject. Again, there is a very interesting article in the journal Blood titled Locus Control Regions, and one can always go through this and try to understand how these locus control regions play a very, very important role in differential regulation of gene expression during embryonic development. Now, I am going to spend maybe a few minutes to tell another important mechanism of epigenetic regulation of gene expression that again has a very lot of implications, that is inactivation of the X chromosome during embryonic development. X chromosome inactivation during embryonic development. Now, as you know, the females usually have 2 X chromosomes, whereas the males usually have only 1 X and 1 Y chromosome. That means genes which are present on the X chromosome, they are represented twice in females, but they are represented only once in case of males. So if you have X-linked genes in the females, the level of expression will be 2 times than the same level of gene expression in the males, which is not correct. That means the genes present in the X chromosome, whether it is male or female, should be expressed only at the same level, right? To attain this, what happens, there is what is known as the X chromosome inactivation. So although the females have 2 X chromosomes, one of the X chromosome is completely inactivated, so that the genes which are expressed in the males and females in the X chromosome are same. the expression of the X-linked genes in the male and females are usually same, because one of the X chromosome females is inactivated. So, women are XX and men are XY in their chromosome composition. All levels, however, the levels of all essential X coded gene products are similar between men and women if women have twice the number of alleles. This was the question Mary Lyon asked us way back in 1971, and what she said discovered is that in cells which contain multiple X chromosome, all but one is inactivated during mammalian embryogenesis, and this is what she called as a Lyon effect. Okay, if you have, for example, 3 X chromosomes, only one X chromosome remains inactive, all others get inactivated. In the females, we have 2 X chromosomes, only one remains active, another remains inactive. Now, the question is, which one gets inactivated? Is the paternal X or maternal X? Again, there are a lot of interesting differences. Usually, whether the paternal X chromosome should be inactivated or maternal X chromosome inactivated, it is a random phenomenon. In some cells, paternal X chromosome get inactivated, in some other cells, maternal X chromosome get inactivated during embryonic development. But in certain mammalian species like marsupials, that is kangaroos for example, it is always the paternal X chromosome which gets inactivated. Whereas in eutherian mammals like humans, in some cells paternal X chromosome gets inactivated, in some other cells maternal X chromosome gets inactivated. In cats for example, again it is random, and that is why you have these variegations. In cats you always have these pigmentation genes, because depending upon what kind of, in some cells paternal X or maternal X get inactivated, that is why you have these varying coat colors. This is the first classic paper which was published by Mary Frances Lyon in 1961, Gene Action in the X chromosome of the Mouse in Nature, which actually threw light upon this differentiated X chromosome inactivation, and the inactive X chromosome can actually be stained as a very dark body, and is often referred to as the bar body. So, if you now do a cytological staining using typical DNA stains, and in the female cells you simply take the buccal smear from a female and buccal smear from a male, in the female buccal smears, if you now stain for DNA, you will find this bar body, because one of the X chromosome is, because it is highly heterochromatinized, it can take up lot more of the stain, and you can see it as a heterochromatinized region as a bar body. Now, what is the mechanism by which this X chromosome inactivation takes place? It turns out, once one of these X chromosome is inactivated, it remains inactivated for the rest of the life. So, this is different from the transient activation inactivation that we normally see for other genes. Many of these inactivations are for the rest of the life, it just does not change. Now, it turns out the last 2 decades a lot of information has come out on the mechanism by which this X chromosome inactivation takes place. It turns out in the X chromosome, near the centromeric region, there is what is called as a XInactivation Center or XIC. Now, this region, which are pointed by arrow here, contains about 7 cogenes which codes for proteins, and 5 genes which codes for RNA, but this RNA does not get translated into proteins. That means 5 non-coding RNAs and 7 protein coding RNAs arise from this XInactivation Center of the X chromosome. This XInactivation Center is very, very crucial for X chromosome inactivation. If you remove this XInactivation Center, then X chromosome inactivation does not take place. And if you take this XInactivation Center and put it on another chromosome, that entire chromosome can get inactivated, clearly indicating that the mechanism by which X chromosome inactivation takes place is this region is responsible for the X chromosome inactivation. Turns out the 2 important genes that play a very, very important role for the X chromosome inactivation are what is called as XIST, pronounced as ZIST, and TSIC or CISC. Now, ZIST and CISC these are 2 genes which are actively involved in inactivating X chromosome. Both of them are non-coding RNAs, they do not code for any proteins. Turns out the X chromosome which remains active, the expression of ZIST is very high. The X chromosome which remains inactive, so I called as XI, the ZIST expression is very high, whereas TSIX expression is very, very low. On the other hand, the X chromosome which remains active, ZIST expression is very low, TSIX expression is very high. So, these 2 transcripts have a reciprocal relationship. And what is this ZIST? Now, the ZIST is nothing but a 17 kilobase long RNA that is expressed on the inactive X chromosome, but not on the active X chromosome. It is processed similarly to the messenger RNAs, but it remains untranslated, and this inactive X is coated with this transcript which is essential for the RNA. So, once this transcript is made from this X chromosome, this RNA coats the entire X chromosome, and therefore, as I showed in this cartoon here, once this XIst RNA is transcribed from the inactive X, this Xist RNA coats the entire X chromosome, and once this X chromosome remains coated with this Xist RNA, it remains inactive, whereas the other allele in which this RNA does not code remains active. So, a non-coding RNA called Xist, which is translated from one of the X chromosome, when it coats the entire X chromosome, that leads to inactivation of the X chromosome. Whereas the activity of the TSIX is reciprocal to that of X and is expressed, the TSIX, that means the chromosome in which the TSIX is expressed, Xist is not expressed, and therefore it remains active. Whereas the chromosome in which Xist is expressed, TSIX is not expressed, therefore it remains inactive. So, depending upon which transcript is expressed, the chromosome either will remain active or remain inactive. Now, what is the mechanism? The mechanism is again simple. Once the Xist RNA coats this entire X chromosome, it now attracts DNA methyltransferases, and these DNA methyltransferases methylate the cytosine residues all along the chromosome, X chromosome, and this DNA methylation now in turn attracts certain chromatin modifying proteins including histone deacylases, and this results in complete heterochromatinization of the X chromosome, and that is how X chromosome inactivation takes place. So, you can see again DNA methylation plays a very important role in the X chromosome inactivation. As I shown here, once the Xist RNA coats the entire X chromosome, which has to be inactivated, it attracts a number of histones as well as histochromatin modifying proteins, and as a result it controls high condensation, therefore becomes heterochromatinized and therefore becomes inactive, and the genes there are not expressed. So, DNA methylation plays very important role not only in imprinting but also in the X chromosome inactivation as well as gene specific regulation during embryonic development. I have given here a number of references. Although I have told you in a very simple manner, X chromosome inactivation is a very complex subject, there are a number of mechanistic issues which are very important, very interesting, and you can read some of these papers to understand how this X chromosome inactivation takes place. Now, I will come to the last stage of my lecture here. Understanding this epigenetic modification during early embryonic development has several implications. To understand how this DNA methylation takes place, how histone modifications takes place during embryonic development, and how differential gene expression and differential chromosome inactivation during embryonic development has number of implications in nuclear cloning, differentiation of embryonic stem cells to specific cell types as well as inducible pluripotent cells. In the next class, we are going to discuss some of these aspects to understand how understanding epigenetics during embryonic development is now paving way for all these studies. Why nuclear cloning has not succeeded to very high levels, why the failure rate is very high in the nuclear cloning. If you take a nucleus from an adult differentiated cell and put it in a unfertilized egg, in enucleated unfertilized egg, and then induce it to develop into an embryo like the famous Dolly sheep, ultimately it is not a very healthy animal. There are problems. That is mainly because the epigenetic programming of this adult cell nucleus is not exactly the same as that happens in the fertilized oocyte and sperm nucleus. The diploid nucleus arising out of the sperm and egg nucleus fusion is not exactly the same as the nucleus of the adult differentiated cell, and this is the reason why offspring arising out of such kind of a nuclear cloning experiments are neither healthy nor the success rate is very high. Similarly, people are now trying to convert stem cells into different cell types. Can I cure diabetes by developing pancreatic cells from a embryonic stem cell? Can I develop cardiomyocytes from a embryonic stem cell? Can I develop neuronal cells to treat diseases like Parkinson's or Alzheimer's from an embryonic stem cells? All this again involves differential gene expression. Can you induce expression? Can you reprogram these adult stem cells or the embryonic stem cell to specific cell types so that it can have number of therapeutic applications? All this again involves differential gene regulation, epigenetic reprogramming, and so on and so forth. So, we will now discuss some of these aspects in the next class, how understanding the basic science in or differential gene regulation during embryonic development has now led to better understanding of these kind of a very important biomedical applications. Like for example, nuclear cloning, like what I told you just now, you enucleate a mature oocyte and introduce a somatic or a embryonic stem cell nucleus, activate the development, and then implant the embryo in a surrogate mother. But the problem is, in such cases the birth rate is very low, and many times the offspring which are born have several developmental abnormalities. Therefore, the donor chromatin which is present in the adult nucleus is not exactly same that of the oocyte nucleus, and that is the reason why it has lot of problems. The take home message I want to give is that there is what is called as transient or the short term gene silencing, permanent or long term gene silencing, and these are all again involves a lot of epigenetic reprogramming. Developmental genes that are needed during later stages of development are transiently held in a repressed state during early embryonic development. This is achieved through short term epigenetic marks such as histone modifications, which can be removed before or within cell division. Like in the last class we discussed about homeotic genes, which have to be expressed at different stages of development. These are all achieved by direct histone modifications. You acetylate or deacetylate histones, and depending upon that you can either activate or repress those genes, but these are all transient. Sometimes you turn on and later you can shut off and then you can turn on again. So these kinds of a transient gene expression changes or gene silencings, when you have to temporarily inactivate a gene at a certain stage of development but can activate later, that usually involves direct histone modifications. But many cases when you want to permanently silence a particular gene, that often involves DNA methylation. Certain genes of genome that are marked with epigenetic information that is stably maintained heritable after many cell divisions, such as imprinted genes, transposons, inactive X chromosome, they require long term silencing, and it has to be sustained throughout the development and the lifespan of an organism. This is generally achieved by DNA methylation, an epigenetic mark that refers to addition of methyl group to the 5th carbon base of cytosine, because DNA methylation provides heritable long term silencing that is crucial for an organism. So DNA methylation leads to long term DNA silencing, whereas direct histone modifications often is involved in transient or short term DNA silencing. So this is one article that I recommend you to read, Active DNA Demethylation, Many Roads Lead to Rome, appeared in the Nature Reviews Molecularology, which very nicely discusses some of these early demethylation methylation changes that takes place during early embryonic development. I will stop here.